Good afternoon. I hope everyone is enjoying their lunch, their pre-lecture lunch. This lecture brought to you unofficially by a French fry purveyor that may start with the letter M. It's my pleasure today to introduce Rodrigo Nicolao. Rodrigo is a PhD student who's based at the Universidade Federal de Pelotas in southern Brazil, in the southernmost state of Rio Grande do Sul. Rodrigo has been with us here at the Fort Worth Botanic Garden in Britt since October of 2023. So he's just about 8 months. He's been with us, doing a lot of travel to various herbaria around the world and working a lot in the lab. So Rodrigo is here thanks to a Sanduíche fellowship that he received from the Brazilian federal government. This is a competitive fellowship that's available to graduate students to do study abroad through the institution CAPES. He plans to graduate, defend his thesis next April in 2025. Before coming here to Britt, he completed his Master of Science in Agronomy in 2021, also at the Universidade Federal de Pelotas. And he received his Bachelor of Science in Agronomy from the Universidade Federal de Santa Catarina in 2019. So we've been very lucky and fortunate to have Rodrigo here working with us. I think everybody who's here today has met and interacted with him. He's really a phenomenal student and just a wonderful person. And so I'm excited to see his talk, and I'm sure you all are as well, on hybridization in potato wild species, Solanum section Petota solanaceae from South America. And he's going to share with us a taxonomic mystery with important implications for both breeding and food security. With that, I will hand it over here to Rodrigo. Thanks.
Hi, everyone. I'm pleased to be here today at the Lunchtime Lecture in Botanical Gardens in Britt. And I would like to talk with you about the hybridization potato wild species Solanum section Petota from South America for a specific taxonomic mystery and implications for breeding and food security. I'd like to introduce myself. As Morgan told me, I'm an agronomist and plant breeding, master in agronomy by UFPEL from Pelotas. I'm currently a PhD student in agronomy with emphasis in plant breeding at the Universidade Federal de Pelotas. Is a public federal funded university, and I do my research in collaboration with Embrapa Clima Temperado. Embrapa Clima Temperado. Embrapa is a Brazilian agriculture research corporation in which aims to develop a technological base for genuinely tropical agriculture and livestock farming in Brazil. His researchers range from genetic crop improvement, including potatoes, and for more efficient and sustainable production systems. And today, I'm here in Britt. I came since October last year to archive my research goals, which are especially to visit the most representative herbaria here in USA and evaluate these collections with conserved potato wild species. And also to do the phylogenomics with the sampling species in collaboration with Texas Tech University. And everyone is know, I think, about the big flood is happening last month in South Brazil, with impacted negatively 600,000 people living into the hydrographic basin. But also this big flood causes a lot of negative impacts to the biodiversity. And today I will separate my talk in 5 points. The first is the introduction. The second is the taxonomic history of this group. The third is reproductive biology and crossing experiments. The fourth is genetic and hybridization, and the next steps of my research. Solanum petota comprises potatoes, the third most important crop on earth. We know that potato are very delicious, is nutritious, and in its tubers contains high levels of nutrients, minerals, vitamins, which is very important for human health and also for food security. This crop contains in its basis 5 domesticated species and 107 wild relatives, which are widely distributed in American continent. And those species is very valuable for crop breeding, especially to maintain the genetic basis of potato crop. And this genetic basis, this genetic variability will help on food security. I think everyone knows about the big potato famine in Ireland in 1845, in which almost mice-like fungi called Phytophthora infestum. No, the common name is potato late blight infested and widespread the Europe, especially Ireland. And this fungus has a potential to damage a whole fields in few days. They damage since from the whole part areas and also degrades the tubers, make it unable to store and even to consume. But that time, the potato crop was reestablished at the beginning of 20th century by the introduction of resistant genes from Solanum demissum. This species was collected in 1896, and this has helped to recreate the potato crop. Solanum section pectota is closely to tomatoes, pepinos, and then peppers too. Who is Solanum malmenum? This is a big mystery. Solanum malmenum is a potato wild relative between the more than 100 species. This species was collected by first time in 1893 by the naturalist Malme at Ijuí, is a city at the Rio Grande do Sul state in South Brazil. And the type was conserved in Berlin, but was destroyed. However, the lectotype is still available in herbarium of Swedish Museum of Natural History in Sweden. But why to study Solanum malmenum? Solanum malmenum is very interesting because it has beneficial traits for breeding, and its taxonomy has been debated for more than 100 years. At the genetic resource lab of Embrapa Clima Temperado, we made different and several activities involving potatoes. Since the herbarium activities, the maintenance of in vitro potato germplasm, DNA extraction, flow cytometry analysis, the maintenance of potato germplasm as tubers under greenhouse conditions, as well evaluated these accessions against biotic stress such as drought and here. To crack this mystery of Solanum malmenum, we retrieved several taxonomic treatments complement these species in American continent since the first publishing of bitter. And then Corel in 1962 made a big work with potato in its wide relatives. Is a wide study, and from Hawkins and Harkins Mendes and Oliveira, Mate Sebastian Barbosa, and Spooner are regional floras. And I will tell you about each conclusion and major findings about each researchers in the later slides. By this time, I visited several herbaria here in Americas, including herbaria of Uruguay, herbaria in Brazil, herbaria in Peru, and here in USA, I visited one of the most representative herbaria in Wisconsin, herbaria of Field Museum, Botanical Research Institute of Texas, and Missouri Botanical Garden. From this source, this visiting, we retrieve a geographical distribution, checked coordinate points, and also use some databases to gather geographical distribution, such as GBIF, a naturalist species linking reflora. And we can find Solanum malmeanum in different countries. They are widely distributed in Southern South America, in countries of Argentina, Uruguay, Paraguay, and Brazil. And it's ranged from sea level up to 765 meters of elevation. And here in red dot are herbarium species samples, and in white, in blue dots represents the samples from herbarium, from germoplasm, sorry. We can find these species in different ecological preferences and habitats. For example, in Argentina, we can find them in eastern subtropical province at grasslands, in Paraguay, in xerophytic vegetation, in chaqueño domain, in mixed forest in Brazil with presence of Araucaria augustifolia, and in Brazil and Uruguay, in Pampean province, where predominantly grasses and brushes. These species flowering and fruiting from April to October, and tuberizes under short and long photoperiods. Now, in this second part, I will talk about the history of taxonomic treatment of Solanum malmeanum. This species gives his new certificate of birth in 1913, when the taxonomic Bitter described this wild potato species as a new species for potato science, and published at the Repertorium Speciarum Novarum Regni Vegetabilis. He characterized Solanum malmeanum as a herbaceous plant with a rosette habit up to 30 centimeters in height. His leaf is blade measuring between 11 to 13 centimeters long, leaflet segments of 8 to elliptic, and its white corolla. Bitter differentiate Solanum malmeanum from other species such as Commersoni and Chacuense from few traits, especially on leaf traits. He differentiated malmeanum to contain more pairs of interstitial segment leaflets between each pair of segments, comparing to Commersoni, and also the corolla of Commersoni to be folded front, while in Solanum malmeanum the corolla is folded back. From Solanum chacuense, bitter differentiate from some calyx traits, especially the Lacinian length. Years later, Solanum Correll studied this potato wild species and treated Solanum malmeanum as a form of Commersoni, because he believes that this species could be arising from a natural hybridization event between Solanum commersoni and Solanum chacuense. However, he differentiated the form malmeanum from the form commersone by some leaf traits, and because the inflorescence of commersone to be more compact than the inflorescence of malmeano. And this is the mystery part 2. Is this a hybrid species between commersone and chaquense? In 1969, Hawkins and Hertz studied the flora of Argentina, Brazil, Uruguay, and Paraguay. And he has other bio about the species limit. He treated Solanum commersone with 2 subspecies, subspecies commersone and subspecies malmeanum, based on morphological characteristics and geographical distribution. The main difference on leaves of subspecies malmeano was the leaves, especially the species, especially because the leaves of commersone has its lateral segments in which it reduce in size abruptly compared to those of malmeanum, and due it to the color of the corolla, in which commersone has its purple corolla and malmeanum has blue corollas. Also, he attested that subspecies malmeanum consist an assemblage of biotype with different geographical distribution, and because he is adapted from a different habitat comparing to the Solanum commersone. The blue dots represent commersone, and the purple dots is the malmeano. In 2004, Menzel Oliveira studied the Solanum from South Brazil. He covered and treated Solanum malmeanum the same way of chaquense as a form of commersone. And Mate Sebastian Barbosa studied the flora of Argentina and treated malmeano as the same way of Hawkins and Hertz, treating as a subspecies. And today, mostly of the works follow Spooner. Spooner made a big systematic study on potato wild relative species from the cover whole continent, and divided his studies in 4 volumes representing the potatoes of North and Central America in 2004, and Southern South America and North of South America. Spooner has a semi-vial of bitter. He treats Solanum malmeano as a specific species, and he attested that the Solanum malmeano can be differentiated by morphological traits from other species such as chaquense and commersone. But one of the problems we find in this species is the presence of diploid plants and triploid plants, where in the triploid plants we can see a more robust morphology in habit, in leaf size, in corolla morphology, and an average of increase in size of the tubers. By this point, we have 3 distinct morphologies of the 3 species, the Solanum chaquense, commersone, and malmeanum. Now we need to know about how this species hybridizes and how the gene flow occurs between them. In the third part, we're gonna talk about the gene banks and crossing experiments. Have you ever wondered about what gene banks does? Well, in a simple answer, gene banks store genes, but not literally. Gene banks store collecting plants and seeds from its natural habitats, and these plants and seeds are considered a big reservoir of genetic variability, in which we're going to make a plant variety to breed unique. Sorry. And this genetic variability is also our foundation to create our future food system. And many genes, or specifically genes, will make a crop plant to be more resilient to climate changes and also to combat pests and diseases and to become more nutritious. And this genetic variability is conserved in gene banks, is safely conserved in cases of natural disasters or for people using. Then if a breeder or a farmer are to respond to some climate emergency, they need to assess genetic variability. And this genetic variability might be the solution for their problems. Then this is the reason we need to conserve and rescue as much as possible of the genetic diversity, because once we lose this genetic variability, the crop is gone forever. But gene banks also do other activities. He collects plants, evaluate, and also evaluate these collected plants. For example, the characterization of reproductive biology, evaluate for drought stress, as well for heat stress, is some climate change problems. And this information will make available for people to use. I visited some gene banks to see plants under growth, such as Embrapa Clima Temperado Gene Bank, Instituto Nacional de Investigação Agropecuária e la Universidade de la República em Uruguay, International Potato Center, and Texas A&M College Station. To search and assess the representativity of Solanum malmeanum at gene banks, we use two main databases. The first is the Allelo, is a gene bank database from Embrapa in Genesis. To compile agronomical traits, I use the National Plant Germ Plant System and literature. Actually, we can find 52 accessions of Solanum malmeanum conserved in germplasm around the world, 11 in Brazil, 6 in Netherlands, 8 in Germany, 2 in UK, and 25 in US. Well, Solanum malmeanum is a great source of genetic variability, especially for those beneficial traits for potato breeding, such as pathogen resistance, tolerance to abiotic and stressors, and also it contains several quality traits in its tubers and leaves. And I will present in the next tables. His is the abiotic stress resistant. So many accessions were rescued from the literature, those database, and are reported to be frost and heat resistant. And also many of them is able to tolerate biotical stress, include bacterial and fungi, also some pests, nematodes, and virus. This potato wild species also contains different quality traits in its tuber, such as good dry matter content, tuber starch granules, reducing sugar, protein, amylose, and glycoalkaloids in its leaves. Well, to understand the evolutionary origin, as well the ways to introduce genetic variability to the potato crop, we need to characterize the reproductive behavior from this from the species. Embrapa clima temperado, as I mentioned, they conserve the three species, Solanum chacoensis, Solanum commersone, and Solanum malmeanum. And then we made some crossing schemes involving 10 accession of Solanum malmeanum with different ploid levels from diploids and triploids, one accession of Chacoense diploid, one accession of Commersone diploid, and two accession of Solanum tuberosum, one breeding line and one cultivar released by Embrapa. And we designed the crosses using all the accessions of Malmeanum with all accession with Malmeanum in two ways. Crossing involving Malmeanum and Commersone, crossings involving Malmeanum and Chacoense, and crossings involving Malmeanum and Solanum tuberosum. The crossing experiments was started by selecting tubers with standardized size. We prepared the greenhouse with pots filled with the commercial substrate, and after plant the tubers, the plants starting flowering in about 2 months. To start the pollination process, we need to emasculate the flowers. We will make a treatment, do it to guarantee they will receive the safe pollen and not be contaminated. We emasculate the plants at anthesis, and then we cover the inflorescence. To collect the pollen which we're gonna use in pollination, we're going to collect the pollen at the anthesis stage. We collect the anthers and process in paper bags. This paper bags will go to a chamber with the air circulated with about 25 centigrade degrees. And this will help to release the pollen from the anthers. This pollen, then we put in a small tubes and conserve for short and long term. We pollinate the flowers on morning after emasculating. Then we protect the bag, the inflorescence, and identify the crosses. In about 30 days, if it's successful, we're going to see the fruit formation finishing. We extracted those seeds and submerged in gibberellic acid to overcome the dormancy. And we saw these plants in plastic box filled with this commercial substrate under greenhouse conditions and made a manual irrigation. And the evaluation was made about 30 days after we saw. This is the summary of reproductive behavior of Solanum malmeanum accessions. We see here the pollen production is present in all accessions, especially in diploids. 2 of them didn't produce pollen, and they are triploids. At control treatment, we cover the inflorescence and don't apply any pollination to see the ability of the plant to self-pollinize in natural conditions. Any of them has its ability to self-pollinate. We tested for pattern orcapy, in which is the ability of a plant to produce fruits without seeds. 8 of them was absent of parthenocarpy, and 1 produced induced fruits, parthenocarpic fruits, and 1 produces spontaneous parthenocarpic fruits. We also evaluated for apomixy, that is ability of the plant to produce fruit with seeds without germination. This have meaning and interesting for plant breeding and crop. Any of them present apomixis. We also evaluated for autogamy by manual self-pollinating each accession with its own pollen. 9 of them was considered self-incompatible, and one we have presented self-compatibility. This self-compatible plant raises fruits with seeds, and those seeds germinated. Also, we tested for allogamy. We pollinate each accession with a bulked pollen of those accessions. 3 of them was considered infertile. Coincidentally, they are triploids. And 7 of them was partial or completely fertile, producing seeds. And those seeds germinated very well. We also test the cross-compatibility behavior with Solanum commersoni and Chacoense, and then with Solanum tuberosum. In this table is the crossing involving Malmeanum and commersoni. Mostly of them was successful. Most crosses produce seeds, and those seeds germinated when commersoni pollinated a section of Malmeanum. And reciprocally, the results were similar. We saw that mostly crossings success producing seeds, and those seeds germinated. This is the characteristics of the seed. The seeds were very normal and plump from the two sides, and those seeds germinated very well, especially in crosses with diploids. We also crossed with Solanum chacoense, but the results were dramatically different. When chacoense was crossed with, when Malmeanum was crossed with chacoense, a few seeds was produced from the both sizes, but only the first cross, the seeds germinated. We can see this is the big difference in number of seeds. When Malmeanum pollinated Chacoense, the seeds was higher in number. They was smaller and one-filled. Any of them germinated. But in another way, when Chacoense pollinated Malmeanum accession, mostly produce up to 3 seeds. The seeds was higher, filled, and those seeds germinated. We can see the seeds germinated from the crosses, and those plants grows and flowering. We also test the crossability with tuberosum, and the crossing was considered incompatible because any seed was raised when we crossed it with the cultivated and the breeding line. And from different way, also, we didn't have success in the crosses. The next step, we need to evaluate the crossability with Commerson and Chacoense, covering the whole distribution of those species, these species. Here are the key takeaway from our crossing experiments involving Solanum malmeanum. We can conclude in which malmeanum is predominantly allogamous species. One self-compatible accession, one plant of Solanum malmeanum was identified as self-compatible. It has evolutionary meaning because depending on the way the plant reproduces, they will determine its evolution and promising development of inbreed line for the diploid breeding. This material is interesting to inbreed them and to develop inbreed lines. Solanum malmeanum is cross-compatible with Commersoni, producing normal, plump, and viable seeds. And the barriers of hybridization between Malmeanum and Commersoni are not completed. As we see, the seeds was unfilled in little number and mostly not germinated. However, this incomplete barrier and is partial compatible is hypothesized to be in reason of 2N gametes presence. And Solanum malmeanum is cross-incompatible with Solanum tuberosa. Well, this fourth part of the presentation, the genetic study and hybridization, since we know the reproductive behavior of Solanum malmeanum, and because he's cross-compatible with the Commersoni and sometimes it's Chacoense, we need to understand the gene flow from between these species, and I will present some previous works on phylogenetics, which will help to get inside on this research. One of the first studies involving immunological studies on potatoes was made in 1965 by Lester. He studies the presence of antigens and found that Solanum malmeanum and Commersoni share the same antigen, while Chacoense present other profile. Then they pointing that malmeanum is closer to Commersoni than Chacoense. Some studies was made in Uruguay. This city and collaborators evaluate the germplasm from Uruguay using different molecular markers systems such as AFLP, RAPD, and microsatellites. They found these species form two clear clusters, in which cluster A represent Solanum commersoni in green, and cluster B represents Solanum malmeanum in blue. A phylogenetic study involved a whole section Petota, evaluated the series using AFLP, and in that time, malmeanum was also considered as subspecies, was treated as subspecies. However, these authors attest that malmeanum and commersoni can be genetically distinguished. And here, where is Chacoense? The hybridization in potatoes. Well, malmeanum remains a taxonomic mystery, and he's hypothesized in which malmeanum originated from a natural hybridization event between Chacoense and commersoni. We know that past climatic shifts and geological events impacted the genetic structure and the occurrence in levels of hybridization and introgression among related species. And is not different in Section Petota, where many authors hypothesize in its studies in which natural interspecific hybridization has been a major evolutionary mechanism. Many authors tell us in which about 26 potato species resulted from hybrid speciation, including 5 domesticated and cultivated potato and 21 wild relatives. In study of Hawkes inheriting of potatoes from Argentina, Uruguay, Brazil, and Paraguay, they interpret 9.5% of wild potato specimens as to be a hybrid. And we know in this section, the barriers of hybridization in which maintain the genetic structure are external and internal. The external barriers are spatial, geographical distance, mountains, river, and also temporal, the flower season. We can see, however, that those 3 species coincide in its peak of flowering, and they are mainly pollinated by bumblebees. They can fly up to 2,500 meters of distance. The internal barriers of hybridization resist on this style, in which they produce sRNAs that inhibits the growth of genetically matching pollen tubes, and they can prevent self-compatibility and cross-compatibility. And the podzygotic barriers prevent the apart species to cross each other, and is controlled by the endosperm balance number theory, and this explain a spontaneous abortion of embryo under incompatible crosses. Well, the ongoing studies is that I tell you that Embrapa contains about 18 accession of potato wild species from those 3 species, in vivo and in vitro conserved. Then we are made in the characterization of these accessions by morphological descriptors, morphometrics, flow cytometry in collaboration with Universidade Federal de Juiz de Fora, and an analysis of microsatellites with collaboration with UDELAR of Uruguay, Doutora Paula and Pablo. And also we made different field trips, field works in Brazil to collect those species in its natural habitat. These are some pictures of our expeditions, the materials. We look in the plants, our friend in coastal Brazil. We are processing the samples, and we found them in different habitats and conditions, like in close to the fields, in degraded areas, close to the in border of the roads, into the cities, and the coast of Brazil, South Brazil. This is the picture of the 3 species collecting different times. This is Solanum chacoalense collecting in November, Solanum commersoni collected in June, and Malmeanum collected in October. I think this date is okay. From now, I'm reviewing the geographical distribution of these 3 species to understand the role of geography on diversification of Solanum malmeanum, and doing the characterization of herbarium specimens and the artificial hybrids in which we generate in crossing experiments involving those 3 species, using morphological descriptors, morphometrics, and universal loci from the angiosperms 353 probe set in collaboration with BRIT and Texas Tech University. I will present the morphological exploitation from the species at the botany next week in Grand Rapids. Contemplate the whole distribution of the species in which we evaluate in herbaria. And the next steps from now, I extract DNA samples from those individuals, contemplating a whole distribution, and we submitted to Texas Tech University. We are waiting the sequencing results from angiosperm 353 probe to analyze this data and try to solve a part of this big mystery. And I will present this work in IBC in Madrid in July. And then I will come back to Brazil to defend my thesis until April 25. Then I would like to thank you all for your patience, especially for my advisor, Dr. Gustavo Hayden, Dra. Caroline Castro, and Dr. Morgan Gostel, the CAPES, UFPEL, Embrapa Clima Temperado, BRIT, Texas Tech, IAPT, ASPT, and BSA.
If you have a question, please use the microphone so that those attending on the recording can hear it.
You did a morphometric comparison of the different taxa along with ecological habitat differences. What were the chromosome counts? What's the chromosome diploid number for Solanum tuberosum and the other taxa that are involved here?
Mhm.
And did you look at any pairings of the chromosomes? Why wasn't there any chromosome counts done?
I think I understand. Thanks. Mostly while the relatives, 70% are diploids and triploids, sometimes tetraploids, but mostly are diploids. And the Solanum tuberosa, the cultivated crop, are tetraploids. They are incompatible. But what I did was to explore the three species from contemplated whole distribution, and I evaluated, I think, 52 traits by morphometrics. And yes, I think these traits are expressed by the action of some genes, but is wholly distributed in his genome. I still don't have this information on which chromosome, but potatoes has a number of 24 in diploid levels and 48 in tetraploids level. And Solanum tuberosum, the cultivated crop. In, I think so. They becomes more complex with the polyploidization. True.
Rodrigo, that was an excellent presentation. Thank you. You did a very good job of presenting the literature and what was known to date, and then how your work fit in with it. So well done. I applaud you on a great presentation. This is not a question directly about your research, just I noticed you were talking about the accessions of Malmaeum in gene banks and the number of accessions in the USDA, what was it, the NPGS, was higher than everyone else. Is that a result of David Spooner doing a lot of work and investigation in the new publication, or?
I think those accessions mostly represent the collection of Spooner. And Spooner studied the flora of Argentina. These species occur in Argentina too, and probably they were collected in that country and make a transfer of genetic resources, transfer between countries. And was in, I think it too, because in Argentina was more explored about the potato other relatives comparing to Brazil. Is my like.
Thank you very much.
Thank you, Rodrigo. I just want to say again, great job. That was, I believe, your first hour-long seminar presentation in English. Is that correct?
Exactly.
So just another round of applause really quick.
Thanks.
Really well done. Thank you. I just have a quick question. It's something I haven't thought of, but and I haven't even thought to ask you during your 8 months working here with me. But you got a chance to visit a lot of herbaria during your time here, and you collected a lot of data, both morphological as well as phenological and ecological when you visited the herbaria about specimens corresponding to these three species. You also sampled material that you could use for the genetic analyses that you were able to bring back here. But I was curious, when you were looking at these specimens in the herbaria, have you looked at the timing of flowering in these specimens? I saw that the three species align their peak flowering time in April, May, more or less, it looked like. Have you looked to see if that peak flowering time, the phenology, historically has shifted in these three species from the collections that might go back to the late 19th century all the way up to the present day? Has there been any change that you've observed yet over that historical record of specimens in their flowering time?
No, I didn't yet, but it's a good insight to and a good way to explore, a good data to explore. I didn't, I just exploring in the whole collection, but I didn't do this tiny evaluations. It's a good idea. I will put in my notes.
Great. Yeah, it literally sometimes just
Yeah.
watching
Yeah.
a presentation, even though we've worked so closely together the
Yeah,
past 8
it's good.
months, pops into your head when you see the
It's
data.
good.
Great, thank you.
There's some climate. Yeah, me and Kay talking about the geographical distribution. Me and Kay was exploring those geographical distribution, and we had some previous results to see the potential areas of occurrence from these three species, and we are evaluating for futures scenarios. It don't look so good, but we didn't finish to explore, and we also look back like past scenarios of. Then this is good to put together. Thanks.
Great, thank you. I think there was one other question, but that makes me curious about a follow-up. Maybe I'll ask you afterward.
Mhm.
I'll bring this to Kay.
Yeah, Rodrigo, great presentation. I'm so excited to actually get the context behind. We've been working on this modeling for quite some time now, so getting to actually learn about the biology of these plants is really exciting. So thank you so much. I am curious. You presented a graph about kind of some of the biotic factors that some of the Malmianum lines they're more resistant to heat or to frost. And I saw that a lot of them were frost tolerant, and only one of them showed up as being particularly heat resistant. And I know from our preliminary results with our future modeling, it seems like their populations decline quite a bit in future scenarios. Does that, you think, have to do with some of that? They're not as heat tolerant, and as climate warms, then they're not potentially going to be doing so well in the future. Have you thought about it? I don't know. There's just something that I noticed.
Yeah, that was a good observation, but we didn't evaluate the metrics to assess the representatives in indices, but this tell us we need to try to find and rescue those species, these plants in areas with the heat will exterminate those population. And yes, I think so. Yeah, this.
Yeah, thank you. And I'm also curious, so
I think.
because the crosses were incompatible with tuberosum, is that where kind of like the agricultural crop improvement side stops with Malmianum, or is there some way still that potentially the beneficial traits of Selenium Malmianum can contribute still to crop improvement, even though they are incompatible?
Yeah. Yeah, interesting. Today in potato breeding, there is two ways to create new crops, new potato crops. One is bred in tetraploid levels, propagated by tubers, and the second is the production of botanical seeds in breeding at diploid levels. Those species are separated, classified in processability groups, three main gene pools, and to cross, if you want to transmit genetic diversity from Malmianum to the Solanum tuberosum crop at tetraploid level, we can duplicate the chromosome and use the colchicine into to make it then equal. Or we can reduce the genome of tetraploid tuberosum, make it dihaploid, and then cross with the diploid ones. And also, non-reduced gametes allows to intercross different potato species groups due to transmit the genetic variability using bridge crossers. Like, yeah, there is some species in literature which are used to do this process.
That's really interesting.
Yeah.
I have no idea about the agricultural crop improvement side of things, so that's really cool. Thank you so much.
Crazy biotechnology involved in making a different species diploid or making diploid in order to cross them. But yeah, you can artificially make them compatible. So all of this not necessary. But no, exactly.
Yeah. Yep.
Well, I've got a general question about flooding.
Here, please wait.
I've got a general question about ploidy level. You talked about diploids, you talked about triploids, and I think you even had tetraploids. So my question is, since a tuber is an underground rhizome, do you see any size differences in the tubers based on the chromosome counts?
Yeah, the main difference is the increase in size, and that can be correlated with the chromosome number.
That figures.
Yeah, that in nature we can see, yeah, sometimes in diploid comparing to triploids or pentaploids, you can see a increase in vigor of the plant members, especially the tubers. But mostly, yeah.
Then how did they get the increased size of the domestic Solanum tuberosum and the tubers? Did they select for ploidy level and then plants that exhibited these enlarged tubers? Because the original Solanum tuberosum came from, I think, Colombia, the highlands. It was definitely South America, and then it spread throughout the world.
Mhm. By that time, in the early time, when they started to explore, they take samples from South America and visually they select the tubers by selecting and planting again. But today in the genomics era, we know about there is some genes which are responsible to express this tuber size and different plant traits like leaves or habits, and those genes can be manipulated and
Okay.
controlled in to express more the tubers, for example.
Well, there obviously has been a lot of selection over time for plants that have larger and larger tubers since they're edible. You never said anything about the alkaloids in the Solanaceae or in Solanum, because the above ground points are deadly poisonous, aren't they?
Yeah, the glycoalkaloid was on
That's right.
Yeah, was on the first, in which the first trait in which was negatively selected. The people eat, for example, in Peru, 8,000 years ago, they saw collect the potatoes, start to eat, but the bitter ones they discard and keep selecting the
Right.
bigger ones, less bitter, and this the mastication advance.
So I assume all these plants, if you excoriate the stems or the leaves, they bleed like all the other solanums. Is that true?
Bleed, what you mean bleed? Sorry.
Bleed. They exudate.
Excellent. Yeah. Could you repeat the question, please?
Well, the leaves and the stems the floral parts, I assume that if you take a knife and cut them, you get the exudate.
Mhm.
And sometimes it's colored. Is it white? Is it clear? Are there different colors?
I know in potato is one color.
What? Really? That's interesting.
Yeah, there is liquid in between the stem, but if we cut, they is in color, one color, but not so transparent. Yeah, transparent.
Thank you, everybody for coming today. I think we're not going to do a July lunchtime lecture, so check in on our August lunchtime lectures. We are at time. So thank you so much, Rodrigo.
Thanks.